Good morning, students. Today we will start with molecular basis of inheritance. In the previous chapter, we people learned about the inheritance patterns and the genetic basis of these patterns. At the time of Mendel, the nature of these factors regulating the pattern of inheritance was not so clear. Then, over the next 100 years, it was investigated and there was a realization that DNA is the genetic material. DNA is the genetic material in all organisms. In certain viruses, such as AIDS virus and tobacco mosaic virus, which we call TMV, instead of DNA, RNA is found to be the genetic material. Now, what is this DNA and what is RNA? We'll take up one by one. So, what is DNA? We'll discuss first. So, you see on your screens the DNA molecule, right? So, DNA is the deoxyribonucleic acid. It is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. And both DNA and RNA, that is ribonucleic acid, they are the nucleic acids. Why? Because their location is in the nucleus. Again, as far as the location is being discussed, let me tell you that in the prokaryotic cells, DNA is circular and occurs in the cytoplasm as a component of the prochromosome and plasmids, right? But in the eukaryotic cells, DNA is linear and largely confined to the nucleus as a component of the chromosomes. This is what is known as nuclear DNA. A small amount of DNA also occurs in mitochondria and plastids. This is what is known as the organellar DNA or extranuclear DNA. This DNA is circular like the prokaryotic DNA, and some of the viruses, they also have DNA, others have RNA. Viruses have either DNA or RNA as their genetic material. Now, what is the amount of DNA which is present in the cells? The DNA content is almost constant in the cells of a species. In your previous class, you studied about haploid, diploid cells in cell division. So, you know that just before cell division, the DNA amount is doubled. Why so? Because in order to maintain the constant amount of DNA in the daughter cells, and the gametes have half the amount of DNA because they contain half the number of chromosomes also. Clear? So, we come down to the structure of the DNA. And DNA is made up of a polynucleotide chain, not one but two such chains. So, DNA molecule has two polynucleotide chains held together in a ladder-like structure. The sugar phosphate backbones of the two chains run parallel to each other in opposite directions. Each rung of the ladder is a pair of nitrogenous bases, one purine and one pyrimidine, which extends into the center of the molecule. So, here you can see the structure of one such polynucleotide chain of DNA. the diagram depicting it on your screens. This is one polynucleotide chain. Okay. Now what is a nucleotide? A nucleotide is one of the structural components or the building blocks of DNA and RNA. A nucleotide consists of a base that is one of the four chemicals, that is nitrogenous base, either adenine, thymine, guanine and cytosine, plus a molecule of sugar and a molecule of phosphoric acid. So you can see all the three parts of the nucleotide in the diagram. Here one nitrogenous base is shown here. You can see adenine, which is attached to pentose sugar. Here in this case it is deoxyribose sugar, and this in turn is attached with a phosphate group. So these are the three basic components of a nucleotide, and a chain of such nucleotides forms the polynucleotide chain. Do you understand? This nucleotide chain which you saw here, its basic components are nitrogenous bases, pentose sugar and phosphate group. So you see, DNA being the largest biomolecule, it is a big macromolecule, that is what we mean to say. Okay. And you know, it is made up of two polynucleotide chains. And of this I told you that it is having three components, that is the nitrogenous base, pentose sugar and the phosphate group. Now as far as the nitrogenous base is concerned, it may be purine, and purines are adenine and guanine, or it may be pyrimidine, that is thymine or cytosine, right? Now you see that in each chain the phosphate component is carried by the carbon atom at position 5 end of the sugar, as you can see in the diagram. In one nucleotide unit is joined by a phosphodiester bond to the hydroxyl component of the carbon atom at position 3 of the sugar in the next nucleotide unit. So these 3 ends and 5 end phosphodiester bonds, they provide a stiffness to the polynucleotide chain. Now these alternating sugar phosphate sugar components, they form the backbone of the DNA. Here again we come back to this diagram. Okay, the sugar, phosphate and then sugar. Here you see the attachment and the 3 and the 5 end. So on one side the 3 end is free, and on the other end of the polynucleotide chain 5 end is free. Is it clear? So the nitrogenous base molecules, they are joined to the sugar molecules, and the type of bond formation is the glycosidic bonds, and they project into the space which is enclosed in the helix around 90 degrees to the long axis of the helix. This glycosidic bond develops between one carbon of the sugar and the nitrogen at position 1 in the case of pyrimidine base and at position 9 in the case of purine base. So the two deoxyribonucleotide chains, they are held together by the nitrogen bonds. This we'll see in the further diagrams. You see here the nitrogenous bases. Now here these are the different nitrogenous bases. We talk about 4 nitrogen bases, but here we see the 5. So nitrogenous base is the nitrogen containing base, right? And it is an organic molecule with a nitrogen atom that has the chemical properties of a base. The main biological function of the nitrogenous base is to bond with the nucleic acids together. A nitrogenous base owes its basic properties to the lone pair of the electrons of a nitrogen atom. And you see the purines, that is the adenine and the guanine here, and the pyrimidines, that is the thymine, cytosine and uracil. Uracil is found in the RNA. Right? So uracil instead of thymine is seen in the molecule of RNA. Here you can see both the purines and the pyrimidines, and the binding, a purine with hydrogen bond, it binds to a pyrimidine in the antiparallel nucleotide chains. Right? And in its binding, they bind with specific combination, that adenine will always bind with the thymine and cytosine will always bind with guanine. The hydrogen bonds between adenine and thymine are 2 and between cytosine and guanine are 3. Are you clear with it? Then the sugar molecule, which we were talking about. Here I'll tell you about both the molecules, that is the deoxyribose sugar as well as the ribose sugar. You see the difference between these two? They are both pentose sugars, and pentose sugar is a monosaccharide having 5 carbon atoms. But you see that here the hydroxyl group is replaced with the hydrogen. That means that deoxyribose sugar lacks one molecule of oxygen in it. Is this clear? Then next component is the phosphate group. Now this phosphate group is a molecule containing one atom of phosphorus covalently bound to four oxygen residues, two of which may be expressed as the hydroxyl group. They are relatively reactive molecules that readily form phosphoester bonds by the interaction with the hydroxyl groups. We talked about one term, that is nucleotide, and we talk about three components of the nucleotide, that is the nitrogenous base, a pentose sugar and a phosphate group. But there is one more term which is known as nucleoside. A nucleoside consists of only two components, that is the nitrogenous base and the pentose sugar. So a nucleoside is a nitrogenous base and a pentose sugar, whereas when phosphate is attached to it, it becomes a nucleotide. And when number of such molecules are attached to one another, having these three components, that is nitrogenous base, pentose sugar and phosphate, then it becomes a polynucleotide. And that is what we are studying, isn't it? Okay. So these nucleotides, here on your screens there is displayed the different bond formations. That is the nucleotides, they are joined by covalent bonds between the phosphate group of one nucleotide and the third carbon atom of the pentose sugar in the next nucleotide. This produces an alternating backbone of sugar and phosphate molecules. And you know, a nitrogenous base is linked to the pentose sugar by a glycosidic bond that I told you before. This is an N-glycosidic linkage which forms the nucleoside. And we call it, with adenine if it is formed, we call it as adenosine or deoxyadenosine. With guanine it is known as guanosine or deoxyguanosine. Then cytidine or deoxycytidine and uridine or deoxythymidine. Right? These are the names of the nucleoside with the structure which we, basic structure which we studied. So when a phosphate group is linked to the 5-OH of the nucleoside through phosphoester linkage, a corresponding nucleotide is formed. Two nucleotides are linked through 3,5 phosphodiester linkage to form a dinucleotide. And when more nucleotides join to it, it forms a polynucleotide chain. So a polymer which is formed, it has one end free, that is the phosphate moiety at the 5 end of ribose sugar, which is referred to as 5 end of polynucleotide chain, and the other end of the polymer, the ribose, has free 3-OH group, which is referred to as the 3 end of the polynucleotide chain. We can come back to this diagram. Here you see the 3 end chain, and the 5 at the 5 end at the phosphate end. So we call it as this as 3 end, the hydroxyl group end, and the 5 end, that is the phosphate group end. Okay. So the backbone of the polynucleotide chain is formed by two things, that is the sugar molecule and the phosphate molecule. But the nitrogenous bases, they are linked to sugar moiety and they are projecting from the backbone to one side, and when another such polynucleotide chain comes to opposite to it, it forms the rungs of the ladder. If we consider about this, we go back to its history of DNA. The DNA is an acidic substance present in nucleus, and it was first identified by Frederick Miescher in 1869. He named it as nuclein. However, because of technical limitation in isolating such a long polymer in a intact way, the elucidation of the structure of DNA was not clear for a very long period. It was in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data which was produced by Maurice Wilkins and Rosalind Franklin, that they proposed a very simple but the most popular double helix model of the structure of DNA. The most popular Watson and Crick model. So, one of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double stranded DNA, the ratios between adenine and thymine, guanine and cytosine are constant and are equal. This is what is known as Chargaff's rule. In 1950, Erwin Chargaff formulated important generalizations about DNA structure, and these are known as Chargaff's rules. Now, what does it state? According to Chargaff's rule, the purines and pyrimidines are always in equal amount. That means adenine plus guanine, that is the purines, are equal to thymine plus cytosine, that is the pyrimidines. Then the amount of adenine is always equal to that of thymine, and the amount of guanine is always equal to that of cytosine. So this means adenine equals thymine, guanine equals cytosine. Why? Because when pairing occurs, it is always between adenine to thymine, and the number of bond formation is 2 hydrogen bonds, and between cytosine and guanine, and here the bond formation is 3 hydrogen bonds. But the amount of adenine plus thymine is not necessarily equal to guanine plus cytosine. Why? Because different codings will result in different amino acids and proteins. In human DNA, adenine is around 30.9%, thymine is around 29.4%, guanine is around 19.9%, and cytosine is about 19.8%. Next point of the Chargaff's rule was that the base ratio of adenine plus thymine divided by guanine plus cytosine may vary from one species to another but is constant for a given species. So, this ratio is or this is constant for one species but it varies from one species to another species. Now, this can help us to identify a species from the source of DNA. This means it can help us in classification. The deoxyribose sugar and the phosphate components, they occur in equal proportions. Clear? This is about the Chargaff's rule. Okay. So that is what we say, that the base pairing is very important and it is complementary. So now what are the salient features of the double helix structure of DNA? It is made of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. The two chains have antiparallel polarity. It means if one chain has the polarity 5 end, the other has 5 to 3 end, the other has 3 to 5 end. The base in the two strands are paired through hydrogen bond forming base pairs. Adenine forms two hydrogen bonds with thymine from the opposite strand and vice versa. Similarly, guanine is bonded with cytosine with the 3 hydrogen bonds. This is whatever we have studied up till now. So, this means that a purine will always come opposite to a pyrimidine, right. So, this generates approximately uniform distance between the two strands of the helix also. Then the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometers and they are roughly around 10 base pairs in each turn. Consequently, the distance between a base pair in a helix is approximately equal to 0.34 nanometers. The plane of one base pair stacks over another in a double helix. Now, this in addition to hydrogen bonds confers stability of the helical structure. Here you can see clearly the polynucleotide chain with both the chains, that is the double stranded polynucleotide chain. So, if you see in the upper chain the hydroxyl end, that is the 3 end is free, and on the other side 5, that is the phosphate end is free if we start from right towards left, and below from right towards left it is the phosphate end, that is the 5 end, and the other side it is the hydroxyl end or the 3 end. So, you see one end is 3 to 5, other is 5 end to 3 end. So, both are anti-parallel chains, and projecting from this backbone towards the inner side are the nitrogenous bases adenine, thymine, cytosine and guanine, which are bound by the opposite chain nitrogenous bases by hydrogen bonds, and these hydrogen bonds are 2 between adenine and thymine and 3 between cytosine and guanine. Here it is something like this. It is like a spiral ladder with the rungs in between, and these rungs, they are the nitrogenous bases adenine, thymine, guanine, cytosine, whereas the backbone is formed of the sugar and the phosphate molecules. And there are around 10 base pairs in each turn. Now the central dogma. What is the central dogma in molecular biology? Francis Crick proposed the central dogma in molecular biology. This describes the two-step process, transcription and translation, by which the information in the genes flows into the formation of proteins. You see here that DNA can replicate to form DNA, that is the replication. Transcription is the process, we will study it later on, from which due to this process you can say the DNA forms the RNA, and by translation of this RNA proteins are formed. Right? This is the central dogma of the molecular biology. But in some viruses, the flow of information is in the reverse direction, that is from RNA to DNA. Why so? Because in such viruses, RNA is the genetic material. But normally, the information flows from DNA to RNA. DNA can replicate to form DNA. By transcription, it forms RNA. By translation, it will form the protein. Now the packaging of this DNA helix. Now this is a very long chain which is formed. Right? So it has to be compact in a very small cell. So you know the distance between two base pairs is around 0.34 nanometers. But if the length of DNA helix in a typical mammalian cell is calculated, that is simply by multiplying the total number of base pairs with distance between the two consecutive base pairs. So it is 6.6 into 109 base pairs multiplied by 0.34 into 10 raised to power minus 9 base pairs. So this comes out to be 22 meters in length. Now this length is very big than the cell. In the prokaryotes like Escherichia coli, they do not have a well defined nucleus. DNA is it is in the cell and it is being negatively charged, and it is held by some proteins which are positively charged, and this is what we call as nucleoid. The DNA nucleoid is organized in large loops which are held by the proteins. In eukaryotes, this organization is more complex because there is a set of positively charged basic proteins which are known as histones. A protein acquires charge depending upon the abundance of amino acid residues with charged side chains. Histones are rich in basic amino acid residues, lysines and arginines. Both the amino acid residues, they carry positive charge in their side chains. Histones are organized to form a unit of 8 molecules which is known as a histone octamer, and the charge of the DNA is negative. It is wrapped around the positively charged histone octamer, and both these form a structure which is known as nucleosome. This is important from examination point of view. So around a typical nucleosome consists around 200 base pairs of DNA helix which are wrapped around the histone octamer. The nucleosomes constitute the repeating unit of a structure in nucleus which is known as chromatin thread. Thread-like stained bodies, they appear in the nucleus. So it gives the appearance of beads on string structure when we look at it in the electron microscope. So here in your diagram you can see the histone octamer, and this is the core of the histone molecules. DNA which is wrapped around it, because DNA has negative charge and the histone molecules, they have positive charge. So if you want to see the nucleosome core particle of the electron microscopic view, this is the electron microscopic view of the nucleosomes, which shows the beaded structure, beads in a string. Besides the histone molecules, there are certain non-histone chromosomal proteins also present in the chromatin. This structure of the, along with the nucleosomes, that is the beaded structure, requires packaging for formation of chromosome, and for this it requires additional set of proteins, and they are known as non-histone chromosomal proteins. They are required for the formation of compact thick chromosomes during the metaphase stage, you can say. So in chromatin, the proteins which remain after the histones, they have been removed. They are known as the non-histone proteins, and they can be heterochromatin and euchromatin. In a typical nucleus, there are some regions of chromatin which are loosely packed and they are staining lighter in color. They are known as euchromatin, and euchromatin is transcriptionally active chromatin because it is loosely bound, whereas there are certain parts which are darkly staining, and that is known as heterochromatin, and this heterochromatin is inactive. Thank you so much. Stay safe, stay healthy. We'll meet tomorrow.